So I'm going to start here. We see, this is a dendrogram, and we see this quite a bit when we start talking about soil health. And when we look at soils, we have, three major aspects, right? We have the chemical part, the physical part, the biological portion. And typically when we talk about soil testing, most of the soil tests fall under that chemical sphere. I mean, that's kind of been the focus of soil testing for decades, is we're going to look at the chemical portion of it. Now, yes, there's some physical things that are done. Typically, those are in the field or by survey crews. There's not a lot of physical parameters that laboratories measure. That's changing, but they are a little more difficult. And then now we're really starting to talk a lot about the biological portion. This is becoming, the focus, especially surrounding regenerative ag. And so we have to develop tests that we can start to evaluate the biological portion. And we don't want to forget about the other parts. So here you go. Here's a list of some of the tests. We're not going to go through all these tests. Yes?
Sorry to stop you at the very beginning, but I have my own definition of regenerative ag.
Uh-huh.
Please share yours.
Yeah, asking me to share my definition of regenerative ag. You know what? I don't have one. And the reason for that is because I talk to thousands of people every year. And what I find out is every time I meet with somebody and they share their definition, mine changes because it's about those different perspectives and what it means to them. So typically, when I used to do a lot of soil health talks, just in general, not surrounding the testing, I would talk about kind of defining soil health or regenerative ag for yourself, because it should be centered around your goals for your own operation and your why. So everybody has a little bit of a different why are we doing this, and then what are you trying to achieve on your operation. So that's why I don't really have a definition. I apologize. My why, I guess I can give you my why. My why is because I see producers that are generally trapped between trying to decipher a lot of information coming at you from a lot of directions. Whether it's, the university system, the NRCS, I'll start with those two, they're in here. Whether it's your neighbor, whether it's the product company, the fertilizer dealer, the seed dealer, etc. And so you're trying to basically weed through a lot of information. And with regenerative agriculture, almost like anything, when it started to kind of gain speed, I would receive probably 10 to 15 phone calls every week with somebody who had a product that they wanted to sell for regenerative agriculture. A seed treatment, a biological, a new compost, something, and they wanted to test it, which is great, but then what they really wanted is they wanted me to stamp my name on it and endorse it and say, this is what, if you're going to do this, this is what you need to buy. And I don't like that. I don't endorse it. I don't endorse any products. That doesn't mean that I don't like some of them. I just don't endorse them. So what I'm trying to do is to try to provide data and interpretation to basically bridge the gap between the tools and things that are out there and what you're doing on your operation, and try to help guide you, at least give you some suggestions. So that's my why. If anybody's ever grabbed research articles coming out of the university and sat down and tried to read them, how fun is that? You love it, right? For most people, it's a pretty daunting task. Grab a paper on soil enzymology and start reading that. I've taken 10 years of biology classes at the university level, and 90% of what's in that paper is completely over my head because I'm not an enzyme expert. It takes a lot of years for that. And so I try to do what I can and build that network to pull those people together so we can try to whittle it down and make it understandable. And a lot of what I'm going to talk about today probably is still not going to be understandable. That's okay. We're just trying. We're trying to get there. So kind of looking at some of these tests. I mean, we're not going to go through hardly any of these. But, we've heard of things like soil texture, soil structure, porosity. These are physical measures. We start talking about biological things. We're looking at microbial biomass, pathogens. The chemical stuff we're used to, reactions like redox reactions, cation exchange capacity, phosphorus, nitrate, those types of things. But what we really need to do is try to start focusing on tests that bring some of these things together, because what we've done is just kind of look at them as individual components. And when we start talking about soil, we understand that it's an incredibly integrated system. And when we do something on one end, we usually cause a cascade effect somewhere else. And the testing is no different. Phospholipid fatty acids. How does this test work? What is it for? The goal behind this test is to try to evaluate the microbial community in that soil. Now, there are three major techniques that people try to use to look at microbes in the soil. One of them is microscopy work, so staring through a microscope, trying to do culture plating in a laboratory. Most people, when they think of microbiology, that's what they picture. Culture plates, test tubes, stuff growing, and then a microscope somewhere. The problem with that approach, it's got advantages. If you're looking for something very specific and it's something known to science and it's something we can grow in a laboratory, it works because we kind of know the process. The disadvantage is that it's time consuming, it's expensive, and over 90% of the organisms in the soil, we can't actually grow in a lab. We can't isolate them from their environment and they survive. That's why human pathogens are really easy to cultivate in a lab, because we kind of know a lot about the human body, so we can replicate that environment. Replicating a soil environment to grow these organisms is really difficult, because I'm guessing that your soil environment right outside these windows is a whole lot different than the soil environment outside of a door in Orlando, Florida, and probably different than the environment on the south side of this building where the sun is shining. So it's very difficult to do this. So one of the other techniques out there is genomics, genome sequencing. This technique has made huge strides in the last 5 years. It will become very mainstream, I guess, coming on pretty soon. There are a few companies out there that are doing this. So again, that's a wonderful diagnostics tool if we're trying to isolate certain pathogens, if we're trying to isolate certain organisms in the soil that we know. But the approach we're going to take here is called a molecular approach. We are going to focus on the community as a whole. The disadvantage to this is that we cannot be very specific. If somebody asks me, do I have Fusarium disease? PLFA will not tell us, do we have that disease? We have to use other diagnostic tools for that. But here are some of the advantages to PLFA. All living organisms are made up of cells, and in those cells or surrounding those cells is a membrane, and those membranes are made up of fatty acids. And so we are going to use that to our advantage. Microbe, bacteria, fungi, protozoans, you and I, your dogs, your cats, we all have cells and we all have phospholipids. Soil itself does not. Soil I should say dirt. Soil contains life. Dirt is geology, okay? Dirt does not contain fatty acids. So on this test, we're basically extracting fat out of your soil. You ever think about your soil being fat? Well, that's really what we're doing. And the other important thing is that PLFA represents living microbial biomass. And the reason for that is that when an organism dies, a bacterial cell dies, and it usually breaks open, the other microbes surrounding that eat that dead cell, because it's already got all the components that the living cells need. They're in the right form. Fatty acids, these phospholipid fatty acids, contain phosphorus and a lot of carbon, a lot of energy. So they're very desirable in that soil environment. It is a wonderful source for them, and they go for it. So that's important because we have to have tests that actually respond to management or that respond to environment. And so they have to be able to change. They have to be dynamic. That does create problems elsewhere, but if they never changed, we wouldn't use them because you'd never learn anything from them. I mentioned that they are influenced by land management and environment. So that is another important thing. We actually can change them through our actions, not just letting nature change them. So go ahead.
What if this test is representing a problematic soil with maybe diseases or lots of organisms that cause diseases?
Yeah, so how would you use the...
How would you distinguish?
Yeah, so you can't really distinguish disease from, on this test, diseases from good or good from bad. But what you can look at, it's an indirect understanding, is that we're going to look at, and we'll talk about these, some of the ratios of organisms, the functional group diversity, because following those ecological concepts of, a diverse ecosystem, a balanced ecosystem, those are usually the most resilient to disease. So usually the fields that we see disease issues in, they're very dominated by one group or the other. They've got really low biomass to start with, so there's a lot of opportunity for those pathogens to move in. So we're not measuring it directly, we're just kind of making some guesses or some inferences based off what we see. So, yes, sir.
So this is going back just a bit, where you said about 90% of soil organisms can't be isolated in a lab to begin with. But even if you could isolate them, is it fair to say that they wouldn't behave the same in isolation as they would in ecology?
Absolutely.
They exude different organic compounds?
Yeah. So the question was, if for anybody couldn't hear it, is it even if you could isolate all the organisms, would they still behave the same in a lab? Absolutely not. They definitely would not. Microbes have to survive in a community structure because not every microbe can have all the right tools to survive in the environment. So the old adage that one man's trash is another man's treasure is really true in a microbial world. Certain microbes can degrade cellulose, others can't. But then the byproducts of degrading cellulose become food for the next microbes, and so on and so forth, which is why if you pull them out of that chain, the chain kind of collapses. Yeah, and then the other thing is, the analogy I always use is that if you're going to build a building like this, it's really hard to find one person who is truly an expert plumber, an expert electrician, carpenter. You have to bring in all of those people with different tools and skill sets to actually build something. And so that's kind of what that microbial community does. So absolutely, even if we could isolate them, and then the other part of that is, even if we could isolate them, we don't even know what they actually do. Right? What ecosystem services do they provide? And that's the other challenge. Anybody take anybody take biology class? Ever? Anybody stay awake through biology class? I remember this picture because I think it was on the cover of one of my books, and I was really close to it. Like that. So this is the anatomy of a bacterial cell. And it's very complex. There's a lot of different things in here. You got all these fancy terms, rhizomes and, DNA and plasmids and all this stuff. But when we take that down and we say this is just kind of a cross-section of a cell membrane, it didn't really get any less complex. We still have protein channels. We've got all this other stuff going on. So down here, this is the inside of the cell. This is the outside of the cell at the top. And these red dots with these yellow tails, those are the fatty acids. Those are the phospholipids. Everything else in here, here's some cholesterol. My cells would look a lot different than this, by the way. Peripheral proteins, all these different things. But we're not really interested in all of that. We just want to get those fatty acids out of that cell membrane. So if we strip everything else out, this is what we've got. Here, this is a phospholipid bilayer. They call it a bilayer because these heads on the outside are polar. There's water. Water's polar. It's on the inside of the cell. It's on the outside of the cell. Fats do not mix with water. So the fats face each other to the inside. Now, why that's important is because it keeps you, and it keeps the outside of you, not the you. That makes sense? It literally, it separates you from the environment. And as a microbe, they don't have this big, wonderful portal to bring stuff in, like I do. They rely on taking stuff across this cell membrane. So this is what they actually call a fluid mosaic model. Both of these flow across each other, internally and externally. The fats are kind of liquid on the inside to some degree. But that allows things to sneak in through here and make it into the cell and out of the cell. The other way they do that is with those proteins. Well, I got bad news for the microbe, because in order for us to actually measure these, we're going to destroy that cell. So everybody says, well, I'm really concerned about what's going to happen when I pull a sample because, I'm going to kill the microbes. And I said, I'm going to kill them anyway. We expose the soil to organic solvents. We basically dissolve that fat, and we extract that out of the soil. In the laboratory, we use gas chromatography basically to pull all of these fats out, and not all of the fats are the same molecule. They're different sizes, they're different structures. And what we do with gas chromatography is we separate those into those different structures. And then we get this peak on a computer screen, and we quantify the peaks. So we measure the intensity of the peak and we quantify it. And at the end of the day, we add those all together and we get total microbial biomass. Now, when I say total microbial biomass, we're not actually counting the microbes. We're not actually weighing the microbes. We're just using PLFA as an indicator. The more phospholipids we can pull out of the soil, the more microbes you had to start with. That's the relationship. And we also take those individual fatty acids and we categorize them into different, what we call functional groups. So, the way I explain this is, we've all heard that we're really closely related to chimpanzees, genetically. Right? Everybody heard that? Okay, so 99% of our DNA overlaps with chimpanzees. There's 1% that makes us different. So different, in fact, we're a different species, most of us. I'm more like a gorilla. But with microbes, when we start looking at fatty acids, there's a large amount of them that are called ubiquitous, or they are shared amongst all the different groups of organisms. Some of them are just everywhere. But there's a small percentage of them that are unique to certain groups. Arbuscular mycorrhizal fungi have a higher proportion and a different fatty acid profile than bacteria do. So we pull out those unique pieces, we call those biomarkers, and we sum those together and categorize them into these groups. So when you look at that report, you see things like total bacteria, total fungi, protozoans. That's how we're determining what those are. We're just looking for those unique fatty acids. Everything else gets shoved into a category called undifferentiated. We don't know where it came from. We just know it's biological and we know that it's microbial because we're looking at the range down below plants and below humans and deer. So that's what we do know. We also know just that higher biomass and higher diversity are better. That's a general term, but that's because every ecologist has always done an evaluation and they say a healthy habitat has a lot of life and a diversity of life. And diversity is not just, I've got 10 different species out there. I've also got relative abundance of those species in balance with each other. We are calculating this exactly the same way, except I told you we can't identify species. So we treat each of those functional groups as a species. Bacteria is treated as a species. Fungi is treated as a species. And then we take their relative abundance using PLFA to calculate a diversity index. So here's kind of a ranking table. Now, here's the part. No standard ranking established for this. if you ask me, what should my biomass be, I'm going to say, I don't know. Let's see what we can do. This test has been around for decades, but the problem is it hasn't been used commercially. And so what I've done is I've run well over 10,000 of these, and we put together kind of a ranking table. Just looking at what kind of ranges we typically see, what's our average. We did the same thing with diversity. This gives you at least a place to start. And it should be regionally specific, but it's not, because I simply don't have that kind of time and I don't have that kind of data set. If I had 5,000 samples from Boulder, hint, hint. No, I'm just kidding. If we had large data sets, we could create this a little better. But what you can do within your group, you have a wonderful data set to start with. And you can compare and contrast systems. And you can compare and contrast numbers. I will tell you that yesterday I did some individual consults. I actually saw biomass numbers anywhere from 2,000 down in the average range, which is great. I didn't see anything below that, all the way up to 13,000. 4,000 is what I've got on here. The 13,000 was almost like compost. I mean, because I've done compost tests before, and they're that high. So we can take that and kind of establish a range based on management. The reason why there's no standard ranking is because it is completely dependent on soil type, climate, and that thing, all those other things. However, the one thing we definitely do know is that within that within a given climate, within a given basic soil type, and when I say soil type, I mean think larger picture. Most of the soils in this part of the country are very high pH. They're calcareous soils. I'm treating that kind of as an ecoregion soil type, right? They're high clay, most of them. Just like in Nebraska, we would say, well, in the central part of the valley, they're all silt loams. I don't need to break them down into Holdridge silt loam, Hastings silt loam. I mean, we don't have to get that specific, but within a region, they change, but they are strongly influenced by management. The only thing that's different is the potential based on climate. So that makes it a pretty good tool. So when I first get a result back, these are really the two numbers. 90% of what I'm really looking at are these two numbers. If my biomass is high and if my diversity is over average, I don't even look at the bottom half of the first page. And the way I explain that is that this is the report card portion. So if my child comes home from school and hands me a report card and says, here's my report card, I got 3 A's and a B. That's pretty good. Do I need to really dive in and ask them which classes they got A's and B's on? Nope. But if they hand me a report card and say I got 3 A's and a D minus, I want to know where the D minus came from. So I look at the bottom half of that page and I say, okay, my diversity is really poor. What am I missing? If my diversity is high, I know I got everything. But what am I missing if it's low? We look down there and we look at fungi, we look at protozoans. We try to find out what's wrong or what's missing, and then we use what we know about those organisms, what they like, what they don't like, and then we try to alter the management. If one of our goals is to increase that, we alter the management to try to get there. That's how we're using this test. Yes, sir?
All else being equal, these two, how much variation do you get by time of sample?
By time of sample, so like throughout the year? Yeah, that's a great question. So sampling time definitely has an influence. I will tell you, the further north you go in the country, the larger the influence, just because the temperature swings are usually so much greater. The biggest thing, so what we see is we see kind of in the winter time. If we pulled a sample today, we actually did this in Montana. We pulled one every month off the same location, the same field. And what you see is that they follow the growth pattern. The microbes follow the growth pattern of plants. So the plants, this was in a pastured system, so once the plants were active for a couple of weeks, the bacteria started to increase dramatically. And then behind the bacteria came the fungi, behind the fungi came the protozoans. So there's a lag in there of when they're doing that. And you want to just be consistent. So when it comes to sampling, just pay attention to moisture. Just make sure we're not on the excesses. And then also temperature. You want to be at soil temperature. You want to kind of be in that same range. That's a great question. Yes?
I think your next test will answer my question. I was going to ask, are you making the assumption that this high biodiversity means you don't have any nutrient deficiencies, or can you have one with the other?
That's a great question. So there are some people that claim that there's a relation to nutrient uptake and the diversity and all that. That very well may be true, but that's not really what we're trying to say with the test, because we really start to get into, there's so many other things that play a role in nutrient transport and deficiency and all these things. I mean, that's kind of a pretty far stretch, I guess, to really get to, and I don't want to stick my neck out quite that far. I've already got a lot of enemies just because I'm talking about this. I try to leave the fertilizer guys alone sometimes. But it's something to look at, though, because it is true. We see that. In a healthier system overall, we definitely see changes in nutrient uptake. We definitely see changes in what is considered deficient or non-deficient, but they're not really defined yet. We just see it, observations from all over the country at different farms. So that's a great question. Last thing on this is that basically what I do is I rank this biomass. So we'll say biomass, if it falls in here at very good and the diversity falls at average, the overall score of this sample is kind of this slightly above average area. We just rank the two separately and then put them together. I would rather see a sample with lower biomass and higher diversity than the other way around. We have a lot of samples we've run that have been continuous corn for 30 plus years. The biomass is 4,000 and 5,000, but the diversity is less than 1. I mean, corn produces a lot of residue, especially in the eastern part of the country. There's a lot of food there, but you're selecting for organisms that only like corn. And then when you couple that with all the other stuff they put on that corn, you're even narrowing that diversity even further. And so I'm not overly concerned with the biomass part. It's the diversity that's really the more important part, in my opinion. Yes, sir?
When you look at these different categories, bacteria, fungi, protozoa, in terms of this overall ecology, I'm assuming you don't necessarily want to see, I want to see equal numbers of biomass in each, right? They each play their own role.
Right.
But are there standards?
Yeah, so actually you just led me right into this part, which is perfect. Because, I often get asked that question, what should these numbers be? Or should they be the same? Or why is the protozoa number so terribly low relative to bacteria and all these things? So the way we evaluate that is with some of these ratios on the report. The ratios, or the relative abundance of one to the other, is much more important than the actual number itself. Because those numbers are all related to total biomass. And so we can have fungi at 200 on that report, but if the total biomass is 1,000 or 10,000, and that stays at 200, that's a completely different ratio between bacteria and fungi. So the first one here, I've got these 4 listed. There are other ratios on the report. The last 3 or 4 of them are really not geared towards production agriculture. They are really designed more for tightly controlled greenhouse studies, pot studies, things like that with the university systems or other research institutions on an agronomic scale. They're not that important. So I don't have them up here. But the ones I do want to talk about are these 4. The fungi to bacteria ratio. Oftentimes, whenever you talk about soil biology, somebody mentions this ratio. Anybody you talk to, Dr. Elaine Ingham, wonderful soil microbiologist, talks about fungi to bacteria ratio. Dr. Chris Nichols, fungi to bacteria ratio. So if any of those names sound familiar, they talk about this. And like most of these tests, generally the higher the number, the better. But the one thing I will mention is that when we talk about fungi to bacteria ratios, we often hear the term, we want a 1-to-1 balance. If you read articles, popular science articles about the subject, they talk about a 1-to-1 balance. Well, a 1-to-1 balance is not going to happen using PLFA. And it's probably not going to happen in most agronomic soils. But what we do with this, the reason it doesn't happen with PLFA is that bacteria have a higher proportion of phospholipids than fungi do. So if I could collect a pound of bacteria and a pound of fungi, these might have 80% phospholipids, these have 25% phospholipids. So it's not even a fair comparison, pound for pound. So the way we do this is we skewed this report, or this ranking table. So that's the reason why we say good and very good are at 0.3, and not 1-to-1. An average is, kind of slightly bacterial dominated, and that's how we read that. The other thing I'll mention is that you guys and gals are up against kind of an unfair challenge. Bacteria have a natural advantage in high pH soils over fungi. It has nothing to do with your management. It just has to do with where your farm's located and the pH of that soil. Now, that doesn't mean fungi can't survive. It doesn't mean that they won't do well. It just means that bacteria have a slightly higher punch power. The opposite holds true when we get soils down around 5.5 for a pH. Probably not going to find too many of those out here, huh? But when we talk about, where does the largest organism on this planet live? Underground. Oregon. Thank you, Lauren.
I didn't know that answer ahead of time.
Oh, okay. Yeah, it's a fungus in the forests of Oregon. Those soil pHs are down around 5.5, and fungi have a pretty strong advantage there. Plus, they're obviously undisturbed, so it's not in an agronomic system, but they have an advantage. So we can still work with this. And again, if we made this scale regional, we would probably say that if you scored a 0.2, which is average, we would probably say for this region that's probably good. I mean, that's in the good category. That's how we would kind of look at that. But this is to help you track change, because we know that if we reduce the soil disturbance, if we increase carbon, those things promote fungi. At least that's what we've been told, right? If we don't do as much tillage, or we start to reduce fungicide applications, or we start to get more carbon in the system, we can increase soil fungi. Well, we're just trying to show you, is that true or not? Is it working? And that's kind of the idea behind this ratio. The second one here is the predator to prey ratio. This is protozoans to bacteria. Protozoans graze on bacteria in the soil, and the process of doing so helps release nutrients. So bacteria have a C to N ratio on average of around 3-to-1. They're little bags of fertilizer, nitrogen fertilizer. Protozoans, on the other hand, have a C to N ratio of around 15-to-1, again, on average. So when they graze these bacteria, they accumulate a lot of nitrogen. And they got to get rid of that nitrogen. And that becomes waste product. So it's no different than livestock grazing above the surface. They take in things. They create waste products. They contain nitrogen. This is just happening below the surface. Protozoans, now the thing about this ratio, it's always going to be incredibly small, because prey always greatly outnumber predators. At least they should. Right? If the predators start to dominate the landscape, they end up eating themselves out of house and home. So this is Mother Nature's balancing act, but protozoans can graze lots of bacteria in a day. They have to have two major things to do it. They need good soil structure, because they need pore space. They need channels to move through between the pores and get around the soil aggregates to graze on bacteria. And number two, they have to have water to do it. That doesn't mean your soils have to be wet to your naked eye or to the touch, but they have to have water films around these aggregates, around the organic material, so they can swim to feed on those organisms. That makes this test, or this part of the test, pretty challenging and really important going back to the question about time of year and the conditions in which you sample. So it's just something to keep in mind. Now, a lot of these samples I looked at yesterday, that they were taken in the fall, mid to late October, some of them in early September, or in September. But as those conditions, as the soil start to cool down, as things start to dry out, etc., the protozoans just kind of go dormant. If you see a score of 0, it doesn't mean that there's not any protozoans in your soil. Remember, you've got an acre of soil, and we just took a couple of cores. And then we're just looking for a couple specific fatty acids. I mean, it really is a definition of looking for a needle in a haystack. So you don't have a true 0. It just means they're below our detection limit. When conditions are better, those numbers will probably increase. But this is really important for the nutrient part. Gram positive to gram negative, I'm not going to say too much on this other than all bacteria fall into one of these two categories. I want you to understand positive does not mean good and negative mean bad. It has nothing to do with that. All we're looking at here is the balance between these two groups. They both have different adaptations and abilities to survive in the soil. And so we want to know that if you have a good, healthy soil environment, usually these are fairly balanced. If we have big issues with drought or things like that, or excess soil temperature, then we can have gram positives dominate. You're thinking, well, how am I supposed to control the temperature? Well, you do control the temperature on a micro scale in the soil by what you do at the surface. Is the ground covered? Do you have living roots? Is it shaded of any kind? What's the organic matter content? Those things help regulate temperature and moisture in the soil. So it is related to management. Balanced is somewhere between 1 to 1 and 2 to 1, gram positive to gram negative. Gram positives always have a little bit of an advantage typically. The last one here before we jump into the Haney test is saturated to unsaturated fats. How many of you know what saturated and unsaturated fats are? Most people are not reading food labels. Me either. Essentially, that's what we're doing. I told you we're extracting fat out of the soil, right? Well, now we're looking at what portion of that is saturated fats and what portion of it's unsaturated fats. And this kind of relates back to the environmental part. Saturated fats are solid at room temperature, and unsaturated fats are generally liquid at room temperature. If you're a microbe and your entire cell membrane is made out of fats, and you need it to be fluid to get things in and out of the cell, what happens if it gets too cold? If you're all saturated fats, it gels, and now they can't do anything. So they actually change the composition of their membrane based on the environment they're in. And that includes moisture, and it also includes temperature. And again, we can still regulate some of that through management. So this is why we're looking at this. Yes, time of year will change this one as well, but beyond time of year, even within that, you can still do comparisons between management, and that's what we're trying to do here. So unsaturated fats just have a double bond in here. There puts a kink in that fatty acid chain. Saturated just means that there are no double bonds, so everything fits really tightly together. That's why they become solid at room temperature. Okay? So that's it for PLFA. Is everybody still awake? Yeah.
So that in the sample test that you have here, your predator-prey, the result says all prey.
That means that we did not detect protozoans. So all we found was bacteria. We did not detect protozoans in that, so it just says all prey there. You'll never see one that says anything other than all prey or a number, because we'll never have one that says all predators, and you'll never have one that just has nothing in it of any kind. So that's where that came from, though. It's a good question. Did you still have a question?
The ratios, how much stuff do you put in those when you're huge biomass is undifferentiated?
Usually most of the biomass is in the undifferentiated category. That's not uncommon. the ratios, typically when they study these things, so there's like 15 different fatty acids we look for bacteria. And there's like 5 we look for fungi. And there's 5 for protozoans, essentially. And so every time we step down trying to get more specific, we have to reduce the number of fatty acids we're looking at. So if we talk about gram-positive and gram-negative, there's like 8 and 7. And that total is what we have for total bacteria. Actinomyces, there's only a couple. And so that's why we get to a certain level and we can't. That's why the undifferentiated category is generally so high. But odds are those undifferentiated fatty acids are probably coming from both groups, or all 3 of those groups. Protozoans, fungi, and bacteria. We just don't know exactly which one, so we don't want to just split it into thirds and just say this much is here and here and here. But I wouldn't worry too much about it. We just include it because it's part of that total biomass. We don't want to ignore it. Yeah, Ben.
If the functional group diversity index is pretty low, like on that corn example you gave, or say it's higher hayfield that someone's trying to rehab,
Yep.
what would your recommendations be to get that diversity index up higher? Is it as simple as diversifying what you plant?
Yeah. First thing I would do if I could is I would come in, especially like if you're talking about a hay field or a cornfield where probably kind of depleted. Hay, we've been removing stuff for years. I would come in and I would bring in something like manure as an inoculant, but then I would come in with a diverse, I would get a diverse plant community out there right afterwards. Now, I know, that's easier for some people than others. People that are willing to put cover crop mixes in and do some grazing or do some of those things. But that really has been the best way I've seen. It's diversity in plant species and livestock integration. And that doesn't necessarily mean all the time hooves on the ground, but bringing in components of livestock into that system. So if you can bring the manure in, that kind of thing. And yes, I know it's a lot of times it's composted manure. It's sad. It's been aged, but it's still a host of bacteria and fungi in there that can be a part of that. So it's kind of an inoculation, if you will. I do not recommend, however, buying I hope nobody in here is selling this. I do not recommend buying a gallon of bugs in a jug and spraying 9 different species of bacteria at a rate of 1.5 quarts an acre, because even in an incredibly degraded soil system, you're basically adding 1 microbial cell for every 2 billion that are there. And so you end up with, it's like me spitting in the ocean and then telling everybody in Florida to move because the ocean's going to come up. Yes?
For a pasture, if you're going to top dress like that, what's going to get that bacteria to go down where it's less hospitable versus just staying on top where it's all nice?
Yeah, so you're going to lose some. I mean, the ones that are exposed at the soil, and I mean right at the surface of that, but they don't have to be very far into that system. And a lot of those bacteria have the ability to form spores. They form cysts. So as they start to dry out, as they start to, get desiccated or too hot, they just go dormant, and they'll sit there. And the bugs and the worms or anything else that's there that will distribute that manure or distribute that compost, rainwater coming in, you're going to lose billions and billions and billions of them. But the best part is there's still trillions and trillions and trillions of them. So, yeah, that is a good point. I would not incorporate anything. I would let Mother Nature do it.
Is manure the most dense inoculant compared to compost or any other thing?
Is there other inoculants? Yeah, say compost tea. There are some seed treatments that do work. It's a direct seed contact. I just mean broadcasting something like a liquid across an acre is not really a very effective approach. Direct seed treatments do work. We've known that with rhizobiums for years in legumes. There are mycorrhizal inoculants that work well. You just have to be really careful that you're matching up the right inoculant with the right seed, because there are hundreds and hundreds of species of mycorrhizal out there. I've also seen like some things like pelletized manure, or not even just manure, but biomass. There's something out there called prairie food. It's kind of a new thing. and I would encourage you to look at it. I'm not endorsing it, but at least with their focus is right. They're focusing on carbon and focusing on trying to feed microbes rather than just say, here's a microbe in a jug. So there are some things you can do. Like I said, seed treatments do work in certain cases. Once your soil health gets up, though, I really haven't seen any big response from some of those seed treatments. But it takes a while to get up there. Yeah.
So in an enclosed environment like a greenhouse, what would you recommend if you don't have cattle or net rain and things like that to mix your amendments in?
Yeah, so if I was in that kind of a situation, I would still be focusing on carbon. Whether it's mulch, wood chips. I know wood chips are really high seed-in ratio. And again, some of this depends on what the other numbers on your test look like. I wouldn't recommend, something like poultry litter. I would recommend poultry litter in certain situations. I definitely wouldn't in others. If you have high soluble salts, if you have really high phosphorus, sulfur issues, I would not tell you to put that stuff on. We still have to keep track of those things. In some cases, I've actually looked at some greenhouse and garden where the recommendation has been to haul in other soil to dilute out the soil that you have because it's too rich. And that does happen. I've seen organic matter at 14%, soluble salts at 6%. Sulfur at 3,000 parts per million, that kind of thing, and that's part of the issue. But really, it is focusing on the biological part. It's focusing on the carbon input, whatever source you can get to with that. But you choose the carbon input kind of based on those other factors.
We're in such a dry environment. I'm trying to kickstart, like a tired hayfield in the spring. It might be dry for 6 weeks, and that's your manure spreading window.
Well, here's what I would say. Soils are not built from the bottom up. They're built from the top down. So when we build soil, we used to think that we would build it down here, and it would actually, you would build the layers of soil up. And a lot of people think that because if you look at a house, my house I used to live in was built in like 1906, and the sidewalks were 2 inches below the sod layer, the top. Well, it's actually all the biology, the earthworms and those things that bring the mineral portion up to the surface and then mix it with the organic stuff because all the organic stuff's on top. I mean, yes, the roots are there, but I mean, that's where that's happening. So I understand everybody, we want to incorporate. I still think that you're going to be better off by keeping it at the surface. As long as it's not stuff that's going to blow away. That is one of the challenges, but yeah, that's...
So we had a speaker, we had a Soil Revolution conference here in December. There was a speaker from northern Wyoming. He had very dense, heavy clay, and which we have a lot of here, bentonite clay. And it's my aha moment was that it seemed like the clay is so fine that if you have water in the soil, you have no air, and then it dries out and you
get crusts and cracks.
So soil life won't grow either because there's no air because it's wet or because it's so dry nothing will grow. And he was using a yeoman's plow to try to open it up, get soil roots down, and that made sense to me just because you're getting the right conditions for soil life slowly deeper into the soil in that particular soil. Does that make sense to you?
I'm not trying to sound... No. No, it does. It does in the fact that I hear that a lot, and I hear that quite often. But it's understanding what happens with the plow of any kind. I understand we have a soil that's predominant in clay, and I understand the struggle of drying it out, trying to get water into it, but any type of tillage actually perpetuates the separation of sand, silts, and clays, because as the biological portion is in there, we end up with the consumption of the glues that hold the particles together. So the goal should be is to get the aggregates to be built with the clay. And I have seen really well aggregated clay soils in Oklahoma and Texas. It can be done, but as soon as we do the tillage portion of it, we start to break that up.
It's not tilling, it's just kind of a fracturing. You're not turning it. You're just
Right.
allow air and water deeper into the soil.
Well, I would say that if he's able to do it without collapsing that back in, and if it's working, then go for it. But in my experience is that anytime we inject oxygen into the soil like that, and we do any kind of that disturbance, even if you're just knifing something, you're cutting all the fungal networks because they're filamentous. Bacteria don't care. But all the fungal networks, the fungal is what really holds the particles together. Combination with the bacteria producing certain glues, but it's glomalin produced by the fungi that really kind of hold those together. And if we destroy that process, we can really kind of keep that. We're kind of just staying where we are. I understand that, yes, there is some tillage and things that need to happen in order to jumpstart the system, but then as we get things going, we just try to get away from it because you get to a tipping point where you kind of start to go backwards again.
So the regular soils around here are rock hard.
Mm-hmm.
I mean, without doing something, you cannot get a shovel into them.
Question is,
We take the top dress to get 6 inches of friable soil. But when these guys are talking about doing this vertical tilling, when you cut those fungal networks, how fast are they going to rejoin each other? I mean, would it just be a temporary cut and then it's worth it? Versus once they're severed, do you think they don't rejoin?
no, it depends on what else you're doing in the system. Fallow is more detrimental to fungal communities than the tillage part. So as long as you're reducing the fallow part, then they'll rebuild themselves fairly quickly. My question is what made the soils rock hard?
It's archaeology here.
So was it just rock hard, bare soil like hundreds of years ago? I mean, there had to be grass communities. There had to be things, because I go to West Texas every year, and in West Texas they say, look at this. You know what their stocking density rate is for livestock in West Texas? 1.5 pairs per section. Per section. I was on a ranch that was 1 million acres. One ranch. 1.5 pairs per section. That used to be one of the most productive grass areas in the country. They overgrazed it in World War II with sheep, mostly for the wool production, for the uniforms. The wind blows. Does the wind blow here? No, not at all. Wind blows, but it took it in 4 years, and they've basically, does anybody know what caliche clay is? You think you have bad clay, go see caliche. I used a power washer in 6 hours to get an inch of it out from underneath my truck. And I'm not kidding. And it's still in my driveway. That was 2 years ago, and it's still sitting there. It's just part of my life now. But my point is that I understand the challenge. I get what you're saying. I'm just saying that there was something here. Just like down there, there were things growing there, and it was predominantly grass. And now it's creosote bush, catclaw mesquite, cactus, and then nothing in between. And they've lost the soil part. The only place the creosote bushes are this much higher than everything around them. And there's like a little mound of soil there. It looks like a prairie dog mound with a creosote bush growing out of it. Because those roots, that's the only thing that's held the soil there. And the only reason why is because that's the one thing that the cattle and sheep wouldn't eat. That's the one thing they wouldn't touch. And so then the farmers said, well, we're going to get more grass production, so we're going to introduce Barbary sheep out of Morocco. And that's worked real well. They got out of control. Now there's 2.2 million of them running around West Texas. competing for grass on areas where you have 1.5, pairs per section. So you don't have a whole lot to go around. So the only reason I mention that is because I've seen that as well, and it feels like there's nothing there you can do with it. But I'm just saying that I truly believe that the soils are harder, even in central Nebraska. I've got 30-year no-till soils that are about as hard as this floor. No-till, 30-year, hard as this floor. It's not the tillage part, it's a carbon part. Corn, bean, corn, bean, corn, bean. One of them is a carbon-heavy crop. The other one's a carbon-negative crop. We have carbon going into the system 5 months out of the year. We have no carbon going in the system almost, 19 months out of that 2-year oscillation. It's not even a rotation. Yes?
Yeah, I just wanted to say that I have a small lot in Old Town Lafayette where the house was built in 1905. In my basement is dried prairie that it was built on, and so it was never part of a tilled landscape. It was the earlier part of the development there in Lafayette, and I have some really rich soil, clay, high clay, but it's just beautiful soil. I just wanted to say, grasslands, intact grasslands can build some great soil.
Yeah, thank you. You're right, but they're not all created equal, right? Some of them are much more fragile than others, and this would certainly be in a semi-arid, shallow soils, high clay. It'd be very fragile.